Hi everyone, welcome to this talk. So today I want to talk about modulation of entanglements by nucleotide-associated proteins, or NAPs. These are proteins that are abundant in bacterial cells in particular, and they are associated with genome organization and segregation in bacteria. And so it's very important to understand how they behave in an entangled and crowded system that is the cell, the nucleoid in vivo. And so this is the kind of the aim for this talk. Before going into the details of this project, and I'll tell you what are the results of this, the findings of this project, I'll give you just an overview of other things that we are interested in at the moment. So in collaboration with Professor Robertson Anderson, we've been looking at functionalizing solution of DNA, entangled DNA. So for instance, in this particular project, that there are people talking about this in sessions A and B25. We have an entangled system of DNA in which we add a restriction enzyme. The restriction enzyme starts to chop DNA. And we see that depending on whether we start from a linear DNA or a circular DNA, we have two completely opposite responses. We could have that the system becomes thinner, so the viscosity goes down in time, or even if we start from a circular DNA, from a plasmid DNA, we see that the viscosity can go up in certain conditions. And so this is a kind of a very interesting system in which we can modulate the rheology of the entangled solution by using restriction enzymes. A similar, although different system is the one Maria Panocchido will talk about in A25. She will talk about a similar system of entangled DNA in which we use ligase enzymes instead of restriction enzymes. In Georgia and Yair, so Yair will talk about in B25, and Georgia has a poster in this session talking about the DNA nanostar hydrogels, and we're also looking into functionalizing these hydrogels by using proteins. So if you're interested into that, you can have a look. We're also interested in rheology of DNA origami. So Jenny is working using DNA nanotechnology, DNA origami, to fold tadpole shape polymers, and the aim is then to put them in high concentration so that some collective behaviors and hierarchical topological constraints arise, and they should give rise to interesting rheological behaviors. And finally, we're also interested in the topology and the material properties of kinetoplast DNA. So this is quite a popular topic also, especially this year at the APS. You can also look at Biana's talk in Y66, or the theory of knitting section, or Alex Klotz's talks as well. So let's go back to the nucleoid-associated proteins project. So this is a collaborative project with people in York, so Mark Leek, Agnes Noy, and Jamie Howard, and all the simulations that I'll show you today were made by Yair, and the experiments of microbiology that I'll show you today I made myself. So NAPs are, as I said, it's a range of different proteins that do different things to compact, to regulate the genome organization in bacteria. Recently, and this is actually work from last year from the group in York, they found that the IHF not only bends DNA at extreme angles, but in some conditions it can also bridge DNA. So for instance, they saw these aggregates at the single molecule level with single molecule techniques. Another interesting finding is that if you stain mucus from cystic fibrosis patients, for instance, where you stain for DNA and IHF, what you see is that they colocalize in these kind of hubs and networks, which actually are quite important for the biofilm stability, because if you target with an antibody IHF, you see people found that the Young modulus, the stiffness of the biofilm decreases, suggesting that IHF is actually important to stabilize these entanglements. So then the question naturally arises, which is, how does IHF regulate the DNA organization in the cell? Does it remove entanglements, or does it stabilize DNA bridging, like in this and this other case? Most of the experiments up to now to study the behavior of IHF is mostly single molecule techniques, but here we want to really aim to get insights into the behavior in the bulk, a little bit like comparing one spaghetti with a bowl full of spaghetti, which is a little bit more satisfying in some respect. So first, we perform molecular dynamic simulations of dense DNA polymers modeled as coarse-grained polymers made of beads and springs. We add kinks to model IHF, and what we see first of all is that the radius of gyration of the polymers decreases as we add more and more kinks. So this is kind of expected. We can see that the collapse can also be modeled effectively as the polymers becoming effectively more flexible. This is also quite expected because the polymers are more kinked. What is less obvious is that we can connect this effective flexibility to the entanglement length. Why is that the case? Because if imagine that I have a semi-flexible polymer that connects to entanglement points, if the polymer is quite stiff, then you have a certain, let's say, contour length that connects these 2 points. But once that you have a more flexible chain, then you have much more contour length that connects these 2 points. This means that effectively the entanglement length goes up. Now there is a phenomenological formula, this one given by in this paper by Uchida and Everires, and that connects the entanglement length to the Kuhn length, which is basically twice the persistent length. And so the prediction is that this quantity, the entanglement length, should go up as the effective persistent length goes down. So we can measure this entanglement length by doing primitive path analysis. I don't have the time to go into the details of what it is, but basically you fix the points of your chains and you remove all the intra-chain intra-chain repulsion, and you leave the inter-chain repulsion, and you lower the temperature of the system in such a way that the polymers want to minimize their path in between the points, but when they find an entanglement with other chains, they cannot. And so this allows us to measure through this formula here the effective, the entanglement length as a function of different number of kinks that we add in the system. As you can see, this goes up by a factor of 2 almost. Now, more kinked and flexible polymers are also, you expect them to be faster, right? Because they're less entangled. And indeed, this is what we see. So if we track the mean square displacement so we track the center of mass of the polymers, we see that the mean square displacement becomes faster and faster as we add more kinks. And also the diffusion coefficient here, it goes up by a factor of 4. And by assuming that this is a repetition, that they perform repetition dynamics, simply because of the fact that the entanglement length goes up by a factor of 1.7, then we can expect that the relaxation time normalized by the case in which we don't have any IHF in the system should be 5-fold faster, and indeed this is exactly what we observe in our simulations. So this is all well and good in simulations. Now we want to check if this is actually true in experiments. So in experiments, what we do, we do microreology. So we embed passive tracers in a fluid of water, in this case, or entangled DNA, just to show you the difference. These are the same passive tracers, 1 micron beads, and here you can see that they move pretty quickly, so they fuse fast. But here, because they are in a mesh of entangled DNA, they don't move as much. We track the position of these beads, and we can extract the mean square displacement. And using the generalized Stokes-Einstein equation, we can reconstruct also the full viscoelastic spectrum. So now the protocol of this experiment is, okay, so it's pretty simple, right, and straightforward. We start from an entangled solution of DNA. We have the passive tracers. We add the protein of interest, which can, for instance, be IHF, which bends or kinks DNA, and then we have a readout on the change in viscoelasticity of the system of DNA by simply looking at the mean square displacement of these beads. And then I want to stress that we work at a volume fraction of DNA that is 1%, that is very close, it's within the range of what you expect in vivo, actually, if you consider only DNA. Now, the first finding that is quite interesting is that even a small amount of IHF is enough to significantly speed up the motion of the beads. So the control is the blue line here. As soon as we add 1 IHF every 800 base pairs in the system, the mean square displacement is significantly faster. By the time, so this is a factor of 3-fold by basically adding between 50 and 100 IHF dimers per lambda DNA, which is 50,000 base pairs. By the time that we add 1 in 80 base pairs, the diffusion coefficient goes up by a factor of 20. And another interesting thing is that if you do a calculation about how many IHF there are in vivo, then you find that there are 30,000 IHF dimers in stationary phase, which corresponds to 150 base pairs of the E. coli genome, which places the system exactly there, which is quite interesting. Now, What's the relationship with the length of the DNA? Because we use lambda DNA, which is it's a 50 kilobase pairs, which is 100 times shorter than E. coli genome, which is almost 5 millions of base pairs. So we perform the same experiment that we did before, but with different sizes of fragments. What we see is that for lambda DNA we get a speed up of factor of 20, but then for shorter DNA. So this is a lambda DNA that's been chopped up in smaller fragments. We don't see a speed up at all, and this is because the system starts already is not entangled. So even if you add the IHF, it does not do anything to the entanglement length or the entanglement themselves because it's not entangled in the first place. Interestingly, so we can extrapolate our results, which are these, so as a function of the average fragment length, to what we expect for the E. coli genome, which is 5 millions of base pairs. And what we see is that the presence of HF may fluidify the E. coli genome by more than 200 times. So this will be, it's a very interesting speculation that comes out of our findings. So just to conclude, so I showed you that the NAPs are interesting, are proteins that basically modulate the entanglements of DNA. I showed you that microbiology can be used to understand how they work in entangled and crowded conditions, and instead so most of the studies to date rely on single molecule experiments. So it's a different way of studying these proteins. I showed you with simulations that by just simply adding kinks to polymers, you reduce the size, you reduce the entanglements, and the polymers go faster. And we see the same effect in experiments, and we don't see actually any evidence of this bridging that is instead important in biofilms and has been seen recently using AFM. So I'll stop here, and I want to thank who did the work and the collaborators and the funders, and I hope there are many questions. Thank you.